This video is audio content only. It does not contain any video footage or images. If you are expecting visual content, please be aware. This is intentionally provided in an audio-only format. We recommend listening to the audio rather than watching the screen. Thank you for your understanding. Welcome to our exploration of genome architecture shapes adaptation. We often view DNA as a simple code, but the physical reality of the cell tells a deeper story. In this narration, we look past the genetic sequence to discover how the 3D structure of the genome acts as a powerful gatekeeper for evolution. From molecular motors and protected gene blocks to the creative potential of genomic parasites, we will see how life's physical blueprint determines the paths of survival. Let's dive into the sophisticated 3D architecture of life. Chapter 1. Spatial Determinism, the 3D Blueprint of Adaptation. To understand how life survives in a world of constant change, we have to look past the simple sequence of the genetic code and consider the physical reality of the cell. If we were to stretch out the DNA found in a single human cell, it would reach about 2 meters in length. Yet this entire thread must be packed into a nucleus that is only about 10 micrometers in diameter. This is the equivalent of trying to stuff 30 miles of extremely thin, fragile string into a single tennis ball without it ever becoming an unusable tangled mess. The way life solves this problem is through a sophisticated system of three-dimensional architecture that does far more than just store information. This structure acts as a physical gatekeeper, a deterministic force that decides which genes are visible to the machinery of evolution and which are locked away in the dark. However, the genome is not a static library. It is a dynamic machine that must constantly rearrange itself to meet the demands of the environment. To manage this, the cell employs a class of molecular motors known as ATP-dependent chromatin remodelers. These are the heavy lifters of the nucleus. They use chemical energy to physically move, eject, or restructure nucleosomes. One of the most fascinating mechanisms they use is a form of molecular ratcheting. A remodeler will clamp onto the DNA and, through a series of conformational shifts, pull a small loop of DNA toward itself, effectively sliding the histone spool along the thread. This is often compared to an inchworm moving along a branch. By shifting these spools, the cell can expose a previously hidden gene, making it available for the machinery of the cell to act upon. This process of moving DNA past the histone core is not just about turning genes on and off. It is a fundamental part of how organisms adapt to stress. Recent research has shown that when an organism is pushed to its limit, the physical state of its chromatin dictates the evolutionary path of least resistance. For example, when cells are placed under constant DNA replication stress, a condition where the machinery that copies the genome is constantly stalled or damaged, they don't just mutate randomly. Instead, the physical constraints of the genome funnel the adaptation toward specific functional modules. In studies of simple organisms like budding yeast, researchers have observed that when faced with this replication stress, the evolutionary response is remarkably reproducible. The cells tend to adapt by modifying 3 specific areas: the DNA replication machinery itself, the checkpoints that detect DNA damage, and the systems that hold sister chromatids together. Even when the starting populations have different genomic setups, some being single-copy organisms and others having double sets of chromosomes, they all gravitate toward the same architectural solutions. This suggests that the physical layout of the genome creates a fitness landscape, where only a few paths lead to survival. The 3D structure of the genome essentially predetermines which genes are on the table for evolutionary change. As we move up in scale, the architecture becomes even more complex. The beads on a string fiber doesn't just float freely, it folds into massive loops and domains. These are often called topologically associating domains, or TADs. Within these domains, DNA interacts with itself much more frequently than it does with DNA in neighboring sections.
These loops are often maintained by specialized proteins that act like architectural staples, bringing distant parts of a chromosome together. This creates a spatial neighborhood, where a regulatory switch located very far away on the linear DNA string can be physically pressed against a gene's start button.
This spatial organization creates another layer of deterministic constraint. If a gene is located within a highly active, open neighborhood, known as an A compartment, it experiences a high level of transcriptional plasticity. It can ramp its activity up or down quickly in response to environmental cues. These regions are effectively the front line of the organism's response to the world, where the physical layout dictates how rapidly a cell can mobilize its existing genetic resources. The physics of this packaging also influences the very likelihood of a mutation occurring in the first place. We often think of mutations as random accidents, but they are heavily influenced by the local environment of the nucleus. Some parts of the genome are tethered to the nuclear lamina, the sturdy envelope that surrounds the nucleus. These peripheral regions are often the most condensed and gene-poor, and they are replicated late in the cell cycle. Because they are physically isolated and handled differently by the cell's repair machinery, they can become hotspots for specific types of genomic instability. The physical position of a gene relative to the center or the edge of the nucleus can be a matter of life or death when the cell is under stress.
Consider the concept of polymer mixing in the nucleus. At a high enough density, long molecules like DNA should, theoretically, become completely entangled. Yet the cell maintains distinct chromosome territories, where each chromosome stays in its own dedicated volume, like neighbors in an apartment complex who rarely cross into each other's living rooms. This lack of mixing is essential for function, but it also creates a physical barrier to certain types of evolution. For two genes on different chromosomes to evolve a new cooperative relationship, they must overcome the physical distance between their respective territories. The 3D genome is essentially a map of possibilities, and the physical distance between any 2 points on that map dictates how likely they are to interact, mutate together, or be co-opted for a new function. This deterministic nature of the genome is particularly evident during the process of DNA replication. When the cell prepares to divide, it must unzip and copy every single one of its billions of base pairs. This is a mechanical nightmare. The machinery that copies DNA must navigate through the dense forest of histones and architectural proteins. If the 3D structure is too rigid, the replication fork stalls, leading to breaks and rearrangements. Scientists have found that the genome's architecture has evolved to manage this mechanical stress. Certain regions are designed to be evolvable. They can tolerate a high degree of structural change without killing the cell. These regions often act as the laboratory of evolution, where new gene combinations are tested.
In the context of rapid environmental change, such as the shifts brought about by human activity or climate instability, this 3D constraint becomes a defining factor in which species thrive and which go extinct. An organism with a highly plastic genome architecture, one that can quickly reorganize its 3D loops to expose new sets of genes, may survive a sudden heat wave or a change in salinity. Meanwhile, an organism with a more rigid architecture may be unable to access the genetic tools it needs to survive, even if those tools are technically present in its DNA sequence. The sequence is the list of ingredients, but the 3D architecture is the kitchen layout that determines what can actually be cooked under pressure. One of the most striking examples of this comes from the study of invasive species. When a plant or animal is introduced to a completely new continent, it often undergoes a burst of rapid adaptation. While much of this is driven by changes in the genetic code, recent evidence suggests that the architecture of the invasion is often set in stone before the first mutation even occurs. The physical accessibility of certain stress response genes determines which traits can be modified quickly.
If a gene for drought tolerance is physically accessible in the native population, that species is far more likely to successfully invade an arid region. The 3D genome doesn't just react to the environment, it sets the boundary for what is possible.
Even the way we measure this architecture is undergoing a revolution. For decades, we could only look at the genome as a linear string of letters. Now, through technologies that capture the contact frequency between different parts of the genome, we can build 3D maps of these interactions. These maps reveal that the genome is organized like a fractal globule, a specific type of folding that allows for incredibly dense packing while still remaining entirely unknotted. This unknotted state is a physical requirement for life. If the DNA were to become truly knotted, the cell would be unable to unzip it for replication or transcription, leading to immediate cell death. This physical requirement for an unknotted state is perhaps the most fundamental constraint on how genomes can evolve. Any evolutionary change that would lead to a physical knot is a dead end. This leads to a profound realization. The tape of life is not being replayed on a blank screen. It is being played on a physical stage with fixed trapdoors, walls, and spotlights. When a population faces a crisis, whether it's a new pathogen, a warming ocean, or a toxic landscape, the 3D genome architecture determines which genes the spotlight of natural selection will hit. It is a deterministic force that funnels the chaotic randomness of mutation into a structured response. This physical reality explains why we see so much parallel evolution, where different species solve the same problem using the same genetic tools. It's not just that those tools are the best for the job, it's that they were the only ones the 3D architecture left out on the counter. As we look deeper into these physical constraints, we begin to see that the genome is not just a digital code, but a physical material with its own set of mechanical properties. It can be stretched, twisted, and compressed, and each of these states carries biological meaning. The twist of the DNA ladder itself can store energy, and when a chromatin remodeler releases that energy, it can trigger a cascade of gene activity. This biophysical perspective is changing our understanding of how life adapts. We are moving from a view of evolution as a slow accumulation of typos in a text to a view of evolution as the constant remodeling of a high-tech skyscraper. Ultimately, the study of these physical constraints is revealing a new layer of biological law. We are discovering that the 3D genome is a deterministic force that shapes the past, present, and future of every lineage on Earth. It dictates the availability of the genetic toolkit, the speed of the adaptive response, and the very boundaries of what life can become. But what happens when these physical boundaries themselves undergo a massive sudden change? What occurs when the architecture doesn't just shift its loops, but actually breaks and reassembles into entirely new configurations? This leads us to the next chapter of the story, where we move from the subtle shifting of nucleosomes to the dramatic world of large-scale structural variants and chromosomal fusions, events that can rewrite the rules of survival in an instant. Chapter 2: Tectonic Shifts: How Structural Variants Shield Adaptive Traits. While the folding of the genome into three-dimensional loops provides the stage for life's daily operations, there are moments in evolutionary history where the stage itself is physically dismantled and rebuilt. If point mutations are the subtle typos in the book of life, structural variants are the tectonic shifts, the massive rearrangements where entire paragraphs are deleted, duplicated, flipped upside down, or moved to entirely different chapters. These events, which we call structural variation, involve segments of DNA larger than 50 base pairs and can encompass millions of nucleotides at once. Recent research is revealing that these large-scale architectural changes are not just random accidents. They are the primary engines behind some of the most rapid and successful adaptations on Earth, particularly in the context of biological invasions and extreme environmental shifts.
To understand why these structural changes are so powerful, we must first look at the problem of gene flow. In the natural world, populations are rarely isolated. Individuals move, mate, and exchange genetic material. While this keeps a species healthy and diverse, it creates a significant hurdle for local adaptation. Imagine a population of insects living on a specific host plant. Over time, they might develop a unique color pattern that makes them invisible to predators on that specific leaf type. However, if they mate with neighbors from a different plant who don't have those mutations, the beneficial combinations of genes are broken apart during the process of recombination. In the next generation, the camouflage is lost and the offspring are eaten. This phenomenon is known as swamping, where the influx of standard genes from the broader population washes away the specialized tools needed for local survival. The genome's solution to this problem is the creation of protected blocks. When a large-scale structural variant occurs, such as a chromosomal inversion where a segment of DNA flips 180 degrees, it creates a physical barrier to recombination. When the cell tries to align its chromosomes during the production of eggs or sperm, the inverted segment cannot pair properly with a standard non-inverted chromosome. This effectively locks the genes within that inversion together. They are inherited as a single unbreakable unit, often referred to as a supergene. While the structural variant is the physical event, the supergene is the functional unit it creates. Over time, as these blocks are maintained in a population, they leave a distinct signature known as a genomic island of differentiation, a region where the genetic code looks vastly different from the rest of the species because it has been shielded from the mixing effects of gene flow. We see a spectacular example of this in the study of Timema stick insects found in the mountains near Santa Barbara, California. These wingless insects exist in two distinct forms, or morphs, each adapted to a different host plant. The striped morph is cryptic on the needle-like leaves of Adenostoma fasciculatum, while the unstriped morph is perfectly camouflaged against the broad leaves of Ceanothus spinosus. These two plants often grow side by side, yet the insects remain perfectly adapted to their specific hosts. The secret to this precision lies in complex structural variation. By using advanced phased genome assemblies, researchers discovered that these color patterns are governed by massive translocations that have also been inverted. These variants span between 15 and 43 million base pairs and contain hundreds of genes. Crucially, these variants arose independently on different mountains without any exchange of genetic material between the populations. This is evolution repeating itself by using the same architectural trick, grabbing a group of genes, moving them, and flipping them to lock them in place. This complex rearrangement creates a protected zone where the genes for color and other host-specific traits are shielded from being broken apart, allowing the insects to maintain their camouflage even when populations on different plants live only a few meters apart. This architectural strategy is a fundamental driver of how invasive species conquer new continents, helping to solve what scientists call the invasion paradox. Usually, an invasion starts with just a few individuals, which should mean the population has very low genetic diversity and is prone to extinction. Yet, invasive species often thrive and adapt with startling speed. The answer lies in these massive, pre-packaged structural blocks. In global studies of common ragweed, Ambrosia artemisiaefolia, researchers have identified large effect blocks that are involved in parallel adaptation across different continents. Ragweed that invades North America uses the exact same structural blocks to adapt to the climate as populations invading Europe. These blocks contribute to a robust genomic foundation that allows the species to be flexible in the face of new challenges, a concept explored further in the context of plasticity in the next chapter. One of the most dramatic forms of structural variation is the chromosomal fusion. This occurs when two separate chromosomes break and then join together to form a single larger chromosome. When two chromosomes fuse, they fundamentally alter the landscape of recombination. Specifically, the genes that were once at the ends of the chromosomes, the telomeres, suddenly find themselves in the middle, near the newly formed centromere. The centromere is a cold spot for genetic exchange. Because the machinery of the cell must grab onto the centromere to pull chromosomes apart during division, the DNA in this region is kept under tight control, and recombination is suppressed.
By fusing two chromosomes, the cell can effectively trap a set of genes in this low recombination zone. This is exactly what has been discovered in a complex of invasive copods, tiny aquatic crustaceans like Eurytomora caroleae and E. gulfia, which are masters of moving from the salty ocean to inland freshwater lakes. This transition is one of the most difficult physiological leaps in nature, requiring massive changes in how the organism manages salt and water balance. In these copods, the ability to survive in fresh water is linked to chromosomal fusions that brought together distant genes involved in ion transport. By joining genes previously located near telomeres and moving them to the centromeric zone, the genome created a protected block of salt management tools. These fusion sites are now hotspots for selection. When a population moves into a new freshwater habitat, the individuals who carry these fused blocks survive, while those with the ancestral separate chromosomes do not.
The impact of these structural variants is often much larger than that of single nucleotide changes. In neopolyploid crops like rapeseed, Brassica napus, structural variants account for a greater proportion of genomic variation than simple typos. Large inversions, some as massive as 26 million base pairs, have been found to reshape gene expression across the entire plant. The pervasive impact of these variants was specifically demonstrated in the glucosinolate biosynthesis and transport pathway, which is critical for the plant's defense against pests and the quality of its oil. These variants don't just affect the genes they contain, they can alter the three-dimensional interactions of the entire chromosome, changing how distant regulatory switches contact their target genes. This suggests a profound shift in our understanding of biology. We are moving from a gene-centric view to an architecture-centric one. For decades, the focus was on finding the specific gene for a trait. But we are learning that the arrangement of genes is often more important than the genes themselves. An organism might have all the right genes to survive a drought, but if those genes are scattered across different chromosomes and prone to being swamped by gene flow, the population will never adapt. However, if those same genes are physically brought together into a protected block, the population can adapt almost instantly. Structural variation provides a level of trait innovation that point mutations simply cannot match, allowing for the sudden emergence of complex multigene traits.
This architectural protection is particularly vital for species facing rapid climate change or human-modified landscapes. In these high-stress environments, there isn't time to wait for a series of beneficial point mutations to arise. Instead, evolution often relies on standing structural variation, pre-existing inversions or fusions that might have been present at low frequencies in the native range but become the key to survival in the new range. These blocks allow for soft sweeps, where an adaptive trait can emerge from multiple sources at once because the structural survival kit was already available in the genomic deck. The presence of these blocks also challenges our understanding of speciation. When two populations develop different structural variants, for example, if one population has a large inversion and the other does not, they begin to become reproductively isolated. Hybrids between the two groups will have chromosomes that can't pair properly, leading to reduced fertility. In this way, structural variation doesn't just help an organism adapt, it can actually trigger the birth of a new species. The genomic islands created by these variants act as anchors around which the rest of the genome can diverge. As we look across the tree of life, we see that structural variation is a universal theme. Even in the world of viruses, Staphylococcus aureus bacteriophages use rapid rearrangements to stay one step ahead of the host's immune system.
This virus host adaptation demonstrates that structural shifts are a fundamental survival strategy even at the smallest scales of life. Even in our own history, structural variants have played a defining role through the accumulation of segmental duplications, large blocks of DNA that are copied and pasted into new locations. These duplications are a form of copy number variation, CNV, and account for about 7% of our genome. They have been linked to the development of complex traits, including the expansion of the human brain. These core duplicons acted as architectural foundations upon which new human-specific genes could evolve. The realization that genome architecture is a dynamic physical force changes how we think about the future of biodiversity. We can no longer just count the number of species or the amount of genetic diversity in a population. We have to look at the integrity of the genomic architecture. If a population loses its protected blocks, if the structural variants that fuel rapid adaptation are lost due to habitat fragmentation or small population sizes, that species may lose its ability to respond to a changing world, even if its DNA sequence remains largely the same. This physical perspective on evolution also opens up new questions about the limits of adaptation. If structural variants are the key to rapid success, what determines which species can generate them? Why are some genomes prone to rearrangement while others are rigid and stable? While a massive inversion might lock in a winning combination of genes today, it also limits the organism's ability to evolve new combinations tomorrow. It is a form of evolutionary commitment, a bet on a specific survival strategy that may or may not pay off as the environment continues to shift. How do these large-scale structural shifts interact with the three-dimensional constraints of the nucleus to fine-tune an organism's response to the world without collapsing into genomic chaos? Chapter 3: The Architectural Shock Absorber. To truly appreciate how life navigates the challenges of a shifting world, we must distinguish between two very different survival strategies: the slow, generational crawl of genetic adaptation and the lightning-fast, real-time response known as phenotypic plasticity. If genetic adaptation is the process of rewriting the manual over centuries, plasticity is the ability of an organism to change its behavior, physiology, or internal chemistry on the fly without changing a single letter of its DNA sequence. For decades, these two forces were seen as separate, one being the domain of long-term evolution and the other a temporary fix for short-term stress. However, recent research into the three-dimensional architecture of the genome has revealed a profound relationship between them. We are discovering a compensatory trade-off where the physical organization of the genome can actually make up for a lack of genetic variation, acting as an architectural shock absorber that allows life to survive in environments it was never designed for. This interaction is most visible in transcriptional plasticity, the genome's ability to ramp the activity of specific genes up or down like a dimmer switch. For an organism to be truly plastic, its genome must be structured in a way that allows these switches to be flipped easily. This is not a universal trait. Some genes are locked behind heavy architectural doors, while others sit in open, accessible neighborhoods. This spatial reality creates a fascinating dynamic. The 3D organization of the genome determines the plastic potential of an organism, and in many cases, this structural flexibility can take the place of permanent genetic mutations. The most compelling evidence for this trade-off comes from the study of estuarine oysters. These creatures are the ultimate survivalists of the intertidal zone. Because they are sessile, glued to one spot for their entire adult lives, they cannot move to find better conditions when the tide goes out or the water temperature spikes. In extensive transplant experiments, researchers moved populations of these oysters from their native, cooler northern habitats to much warmer southern waters. The results were a revelation in genomic compensation. The oysters that survived this transition didn't necessarily do so because they had better heat-resistant mutations. Instead, survival was dictated by the 3D neighborhoods their genes inhabited. Genes located in the most active, accessible neighborhoods showed the greatest levels of transcriptional plasticity, dramatically shifting their expression to cope with the heat and salinity of the southern waters. Here, the trade-off becomes clear. There is a negative correlation between these active 3D regions and the signals of long-term natural selection. The genes that were the most physically flexible in their 3D organization tended to show the fewest signs of permanent genetic change in their DNA sequence. It is as if the 3D architecture provides so much room to move that the underlying genetic code doesn't need to evolve. The structure compensates for the sequence. Conversely, genes in the closed, repressed neighborhoods showed much stronger signals of natural selection. In those locked-away neighborhoods, the only way for the organism to adapt was to wait for a permanent mutation that could break the architectural seal. The 3D genome acts as a fundamental regulator of the speed of evolution, protecting some genes from the pressure of natural selection while forcing others to change or perish.
The biological significance of this control is seen in the ability of an oyster to close its shell, its first line of defense against predators and desiccation. This response is governed by the adductor muscles, and the regulation of this muscle function is tied to a specific gene known as man-eye. In transplanted oysters, the expression of this gene was found to be under intense architectural control. Its activity was regulated by a distal enhancer, a regulatory switch located approximately 100 kilobits away on the linear DNA string. Despite this vast linear distance, the enhancer is brought into physical contact with the gene through 3D looping. In the stressful southern environment, the accessibility of this enhancer changed, and a specific long non-coding RNA was produced to stabilize the 3D contact. This synergetic interaction allowed the oysters to fine-tune their shell closure response in real time, achieving a level of adaptive precision that point mutations alone could never reach. This compensatory role of architecture becomes even more striking in organisms with virtually no genetic diversity. In the world of eco-epigenetics, researchers study asexually reproducing populations, such as clonal plants or the marbled crayfish, where every individual is a near-perfect genetic twin. Despite this lack of variation, these populations can inhabit vast geographic ranges. They do so through habitat-specific epigenetic fingerprints. Even with identical DNA sequences, individuals in different environments develop distinct patterns of 3D chromatin organization and DNA methylation. These structural marks act as a form of environmental memory, maintained over years. This mechanism allows a single genotype to produce a wide variety of epigenetic ecotypes, populations that look and act differently because their genomes are folded differently. This explains the general-purpose genotype hypothesis, which suggests that some species are successful invaders because their genomes are pre-wired for maximum structural flexibility. They don't need to mutate because they can refold their way out of trouble. This strategy is not limited to complex animals and plants. Even human pathogens exploit these epigenetic fingerprints to adapt to the diverse environments they encounter within a host, using structural shifts to evade the immune system or resist antibiotics. However, this structural plasticity is not a free lunch. There is a point where the trade-off breaks down, leading to maladaptive plasticity. This occurs when an organism is pushed into an environment so alien that its 3D architectural response actually works against it. The dimmer switch might ramp up a gene that is beneficial in the native habitat but toxic in the new one. Because the 3D shock absorber has physical limits, it can only dampen so much environmental stress before the system begins to misfire. When this happens, the genome faces a crisis that can only be solved through genetic accommodation. This is a process where the temporary, structural changes in gene expression are eventually stabilized by permanent mutations. The 3D architecture buys the organism time, keeping it alive long enough for the slow machinery of natural selection to catch up and hardwire the adaptive response into the DNA sequence. Plasticity is the scout that explores the fitness landscape, finding the paths that the rest of the genome will eventually follow.
The way different kingdoms of life manage this structure-function relationship also tells us a lot about their evolutionary priorities. When we compare animals and plants, we see two very different architectural solutions. Animals have generally evolved a checkerboard architecture, where the genome is divided into a complex mosaic of spatial compartments. This checkerboard becomes more pronounced as an organism becomes more complex, allowing for the highly specialized gene expression patterns needed for different tissues. Plants, on the other hand, favor a strategy of global folding. Instead of relying as heavily on a checkerboard of 3D compartments, plants often organize co-regulated genes into physical clusters along the linear DNA strand, using the spatial arrangement of the entire karyotype as the primary driver of regulation. This difference reflects their lifestyles. Animals, which can move, use a highly compartmentalized 3D genome to drive complex development. Plants, which are rooted in place, use a more globally integrated architecture to manage the massive coordinated shifts in gene expression needed to respond to environmental stress. Fascinatingly, this plant-like strategy appears to be a foundational state of life. During human embryogenesis, the developing genome actually begins with a plant-like global folding strategy. It is only as specific lineages emerge and the embryo becomes more complex that the architecture shifts from this plastic, global state to the more rigid, animal-specific checkerboard pattern. This suggests that the plastic, globally folded genome is the baseline for rapid change, while the checkerboard is the specialized tool for stability and complexity. The 3D genome also plays a critical role in managing the cost of complexity. There is a long-standing theory that the more complex a trait is, the more genes and interactions it involves, the harder it is to adapt, because a mutation that improves one part of the system is likely to break another. This is pleiotropy, where one gene affects multiple traits. However, recent large-scale studies of thousands of plant species have challenged this. Researchers identified 108 gene families that act as the primary hubs for climate adaptation across deep evolutionary time. Under traditional theory, these hubs should be the most constrained and difficult to change because they are so central to the organism's function. But from a 3D architectural perspective, it makes sense. These 108 hub families are consistently located in the most accessible, active 3D neighborhoods. Because they are physically positioned to interact with so many other parts of the genome, they have the highest adaptive potential. Their 3D architecture allows them to be fine-tuned in 1,000 different ways through structural shifts, bypassing the cost of permanent sequence changes. This leads to a profound shift in our understanding of what makes a gene evolvable. It's not just about the sequence of the gene itself, but its spatial context, who its neighbors are, how often it makes 3D contact with enhancers, and how easily its local chromatin can be remodeled. A gene with a very rigid 3D structure is an evolutionary dead end, no matter how useful its sequence might be. A gene with a plastic, flexible 3D structure is a fountain of innovation.
The 3D genome is the mediator between the environment and the genetic code. It is the interface where the real-time demands of the world meet the long-term records of the past. When an organism encounters a new stressor, the 3D architecture is the first to respond, shifting its loops and opening its compartments to activate the emergency response. If that structural shift is successful, the organism survives, and the 3D configuration may even be passed down to the next generation as a form of structural memory. If the stress persists, the underlying DNA sequence eventually changes to support this new architecture, cementing the adaptation into the lineage. This compensatory dance between structure and sequence is what allows life to be both stable and flexible. It explains how clones can adapt without mutations and how complex traits can evolve without collapsing under their own weight.
But as we consider these elegant architectural solutions, a new question arises. What happens when the entire genomic deck is doubled or even tripled? What occurs when an organism doesn't just refold its genome, but inherits 2, 4, or 6 complete sets of it?
Chapter 4: The Redundant Engine.
In our exploration of the genome's physical reality, we have seen how its three-dimensional folding and large-scale rearrangements act as powerful filters for adaptation. But there is a phenomenon in the history of life that represents perhaps the most radical architectural shift of all: polyploidy. This occurs when an organism doesn't just shuffle its existing genetic cards or refold its hand, but instead inherits 2, 4, or even 10 complete sets of its entire genomic deck. In the standard biological experience, most complex organisms are diploid, meaning they carry 2 sets of chromosomes, one from each parent. Polyploidy breaks this rule entirely, creating a state of massive genomic redundancy. For decades, this state has presented scientists with a profound evolutionary paradox. On one hand, having extra copies of every gene should, theoretically, act as a heavy brake on evolution, diluting the effect of new mutations and keeping a lineage anchored to its ancestral form. On the other hand, polyploidy is often the engine behind the most dramatic leaps in complexity and the sudden emergence of entirely new biological innovations. To understand how life navigates this paradox, we have to look at the genome not just as a sequence of letters, but as a multi-layered system of backups and experimental laboratories.
To grasp the break side of the paradox, we can look at the speed of adaptation under stress. As we saw in the yeast experiments described in Chapter 1, selection under constitutive DNA replication stress consistently targets specific functional pathways. While the functional targets of evolution remain the same regardless of the number of chromosome sets, the pace of change is dictated by ploidy. When these cells are haploid, a beneficial mutation in any of these modules is felt immediately, because there is no backup copy to mask the effect. However, in diploid or polyploid states, the pace of evolution slows significantly. Because the organism carries a second wild-type copy of every gene, a beneficial mutation in one copy is often drowned out by the standard operation of the other. The selective advantage is effectively halved or even eliminated. In this context, genomic redundancy acts as a stabilizer, a form of evolutionary inertia that prevents the population from reacting too quickly to environmental noise. It is a safety net that protects the organism from harmful mutations, but that same net catches and holds back the beneficial ones as well. This stabilizing force is visible across much of the tree of life. In stable, predictable environments where an organism is already well adapted, polyploidy is a massive advantage. It provides robustness and a generalist ability to handle minor fluctuations. But what happens when the environment stops being predictable? What happens when an organism is pushed onto what scientists call a rudged fitness landscape, a scenario where the old ways of living are no longer viable, and survival requires a massive, radical shift in form or function? This is where the paradox flips, and polyploidy transforms from a brake into a powerful accelerator. This accelerator effect is mathematically grounded in large deviation theory. This theory explains how polyploidy increases the probability of rare, significant genetic shifts that would be statistically impossible in a leaner, more streamlined genome. By increasing the number of genetic trials occurring simultaneously, polyploidy allows a lineage to explore the extreme tails of a probability distribution. It makes the highly improbable sudden leap not just possible, but likely over evolutionary timescales. The key to this transformation lies in the concept of neo-functionalization. When you have only 1 or 2 copies of an essential gene, you cannot afford to experiment with them. Any major mutation is likely to break a critical life function, leading to death. But when you have 4 or 6 copies, the rules of the game change.
The organism can maintain its essential functions using a few safe copies of the genome, while the extra copies are freed from the pressure of natural selection. These redundant sets become a playground for evolutionary innovation. They can accumulate mutations that would otherwise be lethal, slowly drifting into new functional spaces until, by chance, they stumble upon a new ability. We see the spectacular results of this accelerated innovation in the world of carnivorous plants. These organisms represent one of the most extreme adaptive shifts in the botanical world, moving from a life of absorbing nutrients from the soil to a life of hunting and digesting animals. This transition requires a complete overhaul of the plant's physiology, from the development of specialized traps to the recruitment of complex digestive enzymes. Recent chromosome-scale genome assemblies have revealed that this innovation was fueled by widespread and complex polyploidy. The East Asian pitcher plant, for example, is decaploid, carrying 10 sets of its genome. The queen sundew, Drosera regia, is a hexaploid, while the Cape sundew, Drosera capensis, exists in a dodecaploid state with 12 sets of chromosomes. In these plants, polyploidy provided the raw material for a multi-level molecular convergence. Specifically, the cysteine protease gene family underwent massive lineage-specific expansion. Genes that were originally used for basic plant defense, such as those that produce enzymes to break down fungal cell walls, were duplicated through these whole genome events. Because there were so many redundant copies available, some of them were retasked to function as digestive enzymes in the plant's traps. This didn't happen once, but repeatedly across different lineages. The massive redundancy of the polyploid genome allowed these plants to recruit existing tools for a predatory lifestyle without losing the original defense functions. It is a prime example of how polyploidy allows for phenotypic leaps that would be impossible for a more streamlined genome.
However, the transition to polyploidy is often a chaotic event, sometimes referred to as genome shock. When two different species hybridize and double their genomes, a process called alloploidy, the cell is suddenly confronted with two different sets of architectural instructions. This can lead to a massive reorganization of the three-dimensional genome. We see this clearly in the humped bladderwort, a carnivorous plant that has achieved a feat of extreme genome compaction. Despite a history of multiple whole genome duplications, the bladderwort has one of the smallest genomes of any flowering plant. It managed this by aggressively deleting junk DNA and intergenic spacers while retaining a typical angiosperm gene complement. The bladderwort serves as a direct refutation of the C-value paradox, the old idea that polyploidy must inevitably lead to large bloated genomes. Instead, it shows that a lineage can delete its genomic junk while keeping the innovation. This suggests that the accelerated phase of polyploidy isn't just about adding more. It's about a dynamic process of redeploidization, where the genome actively sorts through its redundant copies, keeping the useful innovations and discarding the rest. This process of sorting through redundancy is not random. It is often governed by a phenomenon called subgenome dominance, where one of the parental sets of chromosomes takes a leading role in the new polyploid organism. The dominant subgenome tends to maintain more of its original genes and experiences higher levels of expression, while the submissive subgenome is more prone to gene loss and mutation. This hierarchy provides a structured way for the organism to manage its redundancy. It keeps one set of instructions stable to ensure survival, while allowing the other set to be the experimental branch of the lineage. This biased distribution of genetic information, a form of skewness, is what allows polyploid organisms to harbor beneficial mutations in some copies of their genome without compromising their overall fitness. It makes the rare, significant evolutionary events described by large deviation theory much more likely to occur and take hold.
The power of this redundancy to drive global success is perhaps nowhere more evident than in the history of bred wheat. In just 10,000 years, bred wheat expanded from a small core area in the Fertile Crescent to become a dominant crop across the entire planet. This remarkable success is a direct result of its hexaploid nature. Bread wheat carries 3 distinct subgenomes, which it acquired through a series of hybridization events with wild relatives. This composite genome didn't just give wheat more genes, it gave it a massive reservoir of adaptive potential. By analyzing the genomes of thousands of wheat accessions, researchers have found that polyploidy allowed wheat to engage in composite introgression. It was able to absorb between 4 and 32% of its genetic material from wild populations it encountered as it spread. Because it had so much redundant genomic space, it could integrate these new traits, such as resistance to local pathogens or tolerance for different climates, without disrupting its core identity as a high-yield grain. This allowed for a divergent adaptation, where different wheat varieties could specialize for wildly different environments across continents, all while sharing a common hexaploid foundation.
We see a similar window of opportunity in the animal kingdom. While polyploidy is more common in plants, it has played a critical role in major animal transitions, such as the movement from water to land. This transition was marked by massive turnovers in gene content, with large-scale gains and expansions of gene families associated with metabolism, stress response, and immunity. Interestingly, the genomic paths taken during terrestrialization show a distinct layer of complexity. Research indicates that semi-terrestrial species often evolved convergent functional patterns, whereas fully terrestrial lineages follow different, more specialized genomic paths. In some lineages, like certain fish and amphibians, entire genome doublings provided the buffer needed to develop the complex skeletal and respiratory systems required for life on land. But there is a mechanical cost to this redundancy that the cell must constantly manage. Having extra chromosomes means more work for the machinery of cell division. It increases the risk of chromosomal chaos, where chromosomes fail to segregate properly, leading to disease. To counter this, polyploid genomes often undergo chromosomal fusions. While we have seen fusions act as protected blocks for adaptation in other contexts, in polyploids they serve a primary role in mechanical cost management. By physically joining extra sets together, the cell reduces the total number of independent units it must manage during division. These fusions then create their own protected blocks of genes, further shaping the adaptive trajectory of the lineage. This highlights how polyploidy and structural variation are not separate forces, but are deeply intertwined in the architectural evolution of the genome.
One of the most intriguing findings in recent years is how polyploidy influences the predictability of evolution. At first glance, the metaphors of an experimental laboratory or a playground imply a high degree of randomness and drifting. You might think that having so many extra genes would make evolution more chaotic, as there are more ways for things to change. However, the opposite appears to be true. While the mutations themselves are random, the redundancy of the polyploid genome allows these organisms to repeatedly hit the same adaptive targets. Because polyploid organisms are navigating such complex, rugged landscapes, they are often funneled toward the same functional solutions. Whether it's a carnivorous plant in the Americas or one in Asia, the extra genomic space provides more shots on goal, making it more likely that the organism will find the most efficient path to survival. The playground of mutation is ultimately governed by the deterministic constraints of the fitness landscape. This brings us back to the core of the paradox. Polyploidy is a state of stabilized experimentation. It provides the brake that prevents a lineage from drifting away during good times and the accelerator that allows it to leap across evolutionary chasms during bad times. It is a fundamental architect of biodiversity, responsible for the crops that feed us, the complex plants that fascinate us, and many of the major transitions that define the history of life. But as we look at these massive, multi-layered genomes, we have to ask, what is the source of the noise that polyploidy is so good at buffering? What are the actual jumping elements that create the mutations and rearrangements that polyploidy then filters and stabilizes? The playground of mutation is ultimately governed by the deterministic constraints of the fitness landscape.
This leads us to the final piece of our architectural puzzle.
Throughout this narrative, we have caught glimpses of genomic parasites, sequences that seem to move and multiply of their own accord, often triggered by the very stresses that drive adaptation. For a long time, these were dismissed as junk or selfish DNA, but we are beginning to see them in a completely different light. How do these jumping genes interact with the 3D loops, the structural blocks, and the polyploid redundancies we've discussed to create the final functional masterpiece of the adapted genome?
Chapter 5. From Parasites to Architects, the Drivers of Innovation. In the previous chapters, we have explored the genome as a physical structure, a collection of protected blocks, and a redundant library. But to truly understand the engine of biological innovation, we must look at the most mysterious and controversial residents of our DNA, transposable elements. For decades, these sequences were dismissed as genomic parasites or simply junk DNA. They are mobile genetic elements, often called jumping genes, that have the uncanny ability to copy and paste or cut and paste themselves into new locations within the genome. In humans, these elements are not a minor footnote. They account for approximately 45% of our entire genetic blueprint. This massive presence includes various classes, such as long interspersed nuclear elements, short interspersed nuclear elements, and long terminal repeat retrotransposons. We are beginning to realize that these sequences are not merely hitchhikers. Instead, they have undergone a radical transformation over millions of years, evolving from selfish intruders into the essential regulatory architects that drive the unique traits of every lineage on Earth. To understand how a parasite becomes an architect, we have to look at the conflict at the heart of the cell. When a transposable element jumps, it can be a catastrophe. It might land in the middle of an essential gene, breaking a critical protein and leading to disease or death. To prevent this, life has evolved a sophisticated multi-layered defense system. The cell uses epigenetic silencing, employing tools like DNA methylation and repressive histone modifications to lock these elements away in a state of permanent hibernation. However, this is not a perfect seal. Some elements manage to escape these defenses, and it is in these rare leaks that the magic of evolution happens. When a transposable element lands near a gene, it doesn't just bring junk DNA with it, it brings its own internal machinery for starting transcription. Recent high-resolution mapping of the human genome has revealed the staggering scale of this phenomenon. Researchers have identified over 26,000 transcription start sites that are derived entirely from transposable elements. These are not just random accidents, they are functional alternative promoters that help control when and where genes are turned on. This activity is particularly intense in specific tissues. In the human testis, for example, we see the highest count and greatest diversity of these element-derived switches. This suggests that the genome uses transposable elements as a primary source of regulatory variation, testing out new gene expression patterns in the germline where they can be passed on to the next generation. The way these elements function as architects is tied to their evolutionary age. There is a continuous gradient of promoter strength and precision that depends on how long an element has been part of the genome. Evolutionarily younger elements, such as those specific to primates or hominids, often retain their intrinsic promoter motifs. These motifs, like the Tata box, allow for very focused and robust transcription. They act like high-performance, factory-installed switches that can be immediately repurposed by the host. We see this in the primate-specific elements known as ERV-LMALRs, which harbor core promoter signals that allow them to initiate transcription the moment they arrive in a new location. As these elements age and accumulate mutations over millions of years, their role changes. Older, more divergent elements tend to lose their focused initiation patterns. Their architectural integrity begins to decay, and their transcriptional activity becomes broader and more noisy. Yet, even in this degraded state, they provide the raw material for innovation.
A cryptic motif buried deep within an old silenced element can be exposed by a change in the local chromatin environment, or a single point mutation can suddenly turn a piece of junk into a brand new regulatory switch. This process, known as exaptation, is how the genome turns an ancient enemy into a modern partner.
The impact of this architectural repurposing is seen in some of our most fundamental biological processes. In early embryonic development, the genome must execute a perfectly timed sequence of events to transform a single cell into a complex organism. Researchers have found that in mice, a specific mouse-specific transposable element serves as the primary promoter for a gene that regulates cellular proliferation. This element-derived switch produces a unique version of the protein that is essential for the growth of the early embryo. Without this parasitic sequence, the developmental program would fail. We see a similar story in the human genome, where an ALU sequence, the most common transposable element in our DNA, present over 1 million times, has been co-opted as an alternative start site for an oncogene involved in lung cancer. While this specific case shows the dark side of this flexibility, it also demonstrates the power of these elements to create entirely new isoforms of proteins that the gene can then use for good or ill.
This architectural flexibility is not just a human or mammalian trait. It is a universal strategy for rapid adaptation, particularly in the face of environmental crisis. When an organism is under intense stress, its epigenetic defenses can weaken, leading to a burst of transposable element activity. For a long time, this was seen as a sign of genomic collapse, but we now see it as a form of stress-induced plasticity. In invasive species, such as the copod complexes that transition from salty oceans to freshwater, we see elevated transposable element activity, specifically within the regions of the genome that are most involved in adaptation. These elements act as catalysts for structural rearrangements, inducing the very inversions and translocations we discussed in earlier chapters. They provide the mutational fuel that allows a population to explore new phenotypic spaces far faster than would be possible through simple point mutations.
We see this same pattern in the world of insects. In populations of mosquitoes facing the stress of new habitats and human-made toxins, transposable elements have been found inserted near genes involved in insecticide resistance and immunity. These insertions don't just change the genetic sequence, they reshape the local 3D architecture, creating new regulatory loops that allow the mosquito to ramp up its defenses. In fruit flies infected with pathogens, transposable elements account for at least half of all the mutations that actually change the fly's physical traits. This suggests that the jumping behavior of these elements is a feature, not a bug, of the genomic response to the world. It is a high-stakes gamble. The cell allows a certain degree of genomic instability in exchange for the possibility of a life-saving innovation. The historical foundation for this understanding goes back to the discovery of jumping genes in maize. The variegated patterns of color in Indian corn are the direct result of transposable elements moving in and out of genes during the development of the kernel. While this was once seen as a botanical curiosity, we now know it is a window into a fundamental principle of life. The genome is not a static list of instructions. It is a dynamic, self-reorganizing material. Transposable elements are the agents of this reorganization. They are the reason why the genome is evolvable. By constantly inserting new regulatory motifs, creating new protein isoforms, and triggering large-scale structural shifts, they ensure that the lineage always has a fresh supply of architectural options. This perspective helps resolve the mystery of why complex organisms have so much seemingly useless DNA. If the genome were a perfectly streamlined machine, it would be incredibly efficient in a stable environment, but it would be fragile and unable to adapt when things changed. By maintaining a vast library of silenced transposable elements, the genome is essentially keeping a massive stock of spare parts and experimental switches. It is an architectural buffer that allows for long-term survival.
The junk of today is the regulatory masterpiece of tomorrow. As we look at the interaction between these mobile elements and the rest of the genomic architecture, we see a beautiful integrated system. The 3D loops and compartments we discussed in the first chapter provide the boundaries within which these elements can jump. The structural blocks and chromosomal fusions we explored in the second chapter provide the protected environments where beneficial insertions can be locked in and inherited. The polyploid redundancies we saw in the fourth chapter provide the safety net that allows the genome to tolerate the mutational load of transposable element activity while it waits for a lucky strike. The transformation of these elements from parasites into architects is perhaps the ultimate example of the genome principle. It shows that evolution is not just a process of filtering out the bad and keeping the good. It is a process of creative destruction and repurposing. It is the story of how life takes the most chaotic and dangerous forces within itself and tames them, turning them into the very tools it needs to build something new. But this realization brings us to a final profound question about the nature of our own identity. If nearly half of our genome is made of these once foreign sequences, and if they are the primary drivers of the regulatory innovation that makes us human, then where does the host and the parasite begin? We are discovering that the genome is not a single unified entity, but a complex ancient ecosystem of competing and cooperating parts. Every time a cell divides, and every time a lineage adapts to a new challenge, this internal ecosystem is renegotiated. We are the living result of that negotiation, a functional masterpiece built from the remnants of genomic wars. This brings our journey through the architecture of adaptation to a close, but it opens a new chapter in our understanding of the future. As we gain the ability to not just read these architectures, but to edit them, we are stepping into the role of the architect ourselves. We are beginning to use the very tools that evolution has used for eons: CRISPR-based genome editing, site-specific recombination, and synthetic structural variants, to address the challenges of disease, food security, and climate change. But as we take up the tools of the jumping gene and the polyploid leap, we must ask, do we truly understand the physical and deterministic rules of the stage we are trying to rebuild? What are the long-term consequences of disturbing the delicate architectural balance that has allowed life to survive and thrive for billions of years? We have explored the genome not as a static list of instructions, but as a dynamic 3D machine. From the microscopic folding of nucleosomes to the massive tectonic shifts of chromosomal fusions, we have seen how physical architecture acts as a deterministic gatekeeper for evolution. This structure funnels the randomness of mutation into viable paths, allowing life to adapt through spatial plasticity, redundant polyploid engines, and the creative repurposing of transposable elements. These findings shift our perspective from a gene-centric view to an architecture-centric one, revealing that the physical layout of DNA is as critical as the sequence itself. As we gain the power to edit these structures, we must consider the long-term consequences of altering a blueprint refined over eons. We are the living result of an ancient architectural negotiation, one that continues to define the boundaries of life. Thank you for listening.